Welcome everyone to I think this is the first seminar in the BrIN Learning Series. I want to remind everyone that we are recording today, so I hope that's okay. And I'll just kick us off briefly introducing our speaker, Brian Gardunia. So it is my great pleasure to introduce Brian to this learning series. Brian is currently the cotton product design lead at Bayer Crop Science, and he can explain what that is when the right slide comes along. But before that, I think that's a recent role. Before that, Brian was at the forefront of what in Bayer was called discovery breeding for more than 10 years. And discovery breeding is a group at Bayer that focused on research and validation of the latest advances in breeding methodologies before they're scaled up to production pipelines. So Brian's worked over his time in corn and cotton and soy and wheat and canola, as all of these different crops experimented with everything from genomic selection to ML predictions to drone phenotyping and rapid cycling, gene editing, like you name it, he's tried it. So his experience in the application of breeding technologies and methods is deep and wide. And today he's going to be sharing an overview of Bayer's own modernization journey, both how the company changed its breeding practices with new technologies, but also, I think, new ways of working. So welcome, Brian, and I'll hand it over to you.
Thanks, Younghwa. I'll give an introduction of kind of my background, although Younghwa did a really good job of and a quite complimentary job. The reality of the last 12 years for me of being in discovery breeding mostly has been 12 years of doing things that didn't work. And it's been actually a real privilege to have had the chance and the kind of freedom to try things that were risky and that often did fail. And they didn't fire me. So I consider that a good thing. For my background, I have moved around a little bit in the US. Internationally, I've lived in Nicaragua and Bolivia. Nicaragua for 2 years as a Mormon missionary once upon a time when I was almost a child, and I lived in Bolivia working on quinoa as part of my master's degree. And then I went to Texas A&M and worked on cotton. And then I was a popcorn breeder, and then I came to Monsanto, and they really lured me to Monsanto with this chance to be in this discovery breeding role and to try things with breeding that I had wanted to do and hadn't had the chance to do in my small program there at Ag Alumni. And so at Monsanto, I worked on corn for the first 5 years, and then I worked on a variety of the varietal crops. So I led the discovery teams for cotton, wheat, and soybeans, and then transferred to lead a team that was across all the row crops. And then now my current role is cotton product design, and I'll try to explain what that means. When I tell people in my neighborhood that I work as cotton product design, they think I'm designing t-shirts. So I don't design t-shirts, but that's kind of the new reorg structure of breeding. Essentially, I lead the early part of the breeding program, population improvement, and like early selections and the first part of testing and line development. I put some pictures on here of me in graduate school. The lowest one is in when I was in Bolivia working on quinoa, and then working on cotton. And when I was in my corn program, I put the bottom one to be totally honest. If you ask my kids what I do email and meetings. And unfortunately, that's way too much of my current job. So hopefully with COVID ending, I'll be out in cotton fields again soon. And not just on my computer. When I was thinking about how to do this presentation, I had two kind of restrictions. I wanted to do a good overview of kind of how we try to make decisions and how the kind of breeding programs have changed over time. But, you know, I am coming from a corporate private breeding company, and like I'm a little bit limited on what I can present and say. And if I'd had more time, I would have probably had a more like customized presentation for this group. But so I had to like modify slides that I had already approved by legal to give. And so what I'll try to do is answer these 4 questions about kind of how we're how we work in Bayer. And sometimes my slides I think will be really effective of that, and some of them I know are less than ideal, and I'll try to like explain why I put them in and what they're for in addressing these 4 questions. Because and if there's any questions, or if you can't hear me for some reason, my microphone works only if I stare like exactly at the microphone. So if there's any time that you can't hear me or you have a question, like jump in. It's probably more important to have this to be a discussion than a lecture. And so I'll dive in. One of the challenges on being in a large company and running a large breeding programs is there is a lot of complexity. And unlike when I was at, in the university and I was doing research for my PhD or my master's, in the company, we really do have to keep our focus always on the fact that we're making a product for customers. We're in a competitive landscape. So the how your products are performing and against those competitors, how your customers are going to use them, and understanding those customer needs is something that's really important, even at the like kind of discovery beginning kind of basic science end of what we do at Monsanto and now Bayer. That you always have to keep in mind. And so what I'll try to do is break down like how we think of that complexity and how we try to like keep that focus on customers and on and on the breeding program. So when do we make decisions? So in the breeding program, essentially we're starting with lots of possible entries, and you're making selections at every stage of the program down to when a product is released through a brand to get sold to a customer. And even then at that stage, there's lots of decisions to make at the brand and the commercial stage on where a product is going to be sold. What's the hybrid launch strategy? Do you build up lots of seed really fast? Does it grow slowly? Does it get focused to a specific like region, or does you try to sell a hybrid or a variety in a broad area? The other kind of decision that gets made at that commercial stage for us is which brand does it go to? So some hybrids or varieties will go to a national brand, some will go to a regional brand, and there's a different sales strategy for those. And you have to look at performance differently. So something that performs in a broadly adapted range would be a product that would be the best for a national brand. But sometimes there's things that may be like on a broad level, not quite as good, but really regionally adapted. And that's something that is an ideal thing to license or to sell through a regional brand. And so there's decisions on that commercial end. And then as you move backwards in the breeding program, every year we're making a selection of going from hundreds of millions or hundreds of thousands down to those few products that we're going to advance. So at each, from the early stage, you're sorting through many possible hybrids or varieties. down to thousands, and then finally tens and hundreds. And what you're looking at when it's at the beginning of the program is really broad performance. And then down as we get closer and closer to commercialization, it's regional adaptation, placement, fit, cost of production, trying to understand how well that variety is going to work for the customer and where it's going to work and what it would replace. So we have sometimes very highly performing hybrids or varieties that may make it all the way to almost commercialization that don't get released that year and may have to wait because or get dropped because what the commercial need is something that has good disease resistance or has the hybrid they're going to replace it with may not be that same profile. So it's understanding what the commercial team needs, and you get closer and closer to fitting that commercial profile as we go through the breeding program. The other thing to think about when to answer this question of when we're making decisions is when we start a breeding program, you have a lot of things in the beginning part of the breeding program. And I think of it as a funnel. For a hybrid program, you have kind of two funnels. You have a funnel to select inbreds, and then you have a second funnel to select the ideal hybrids. So you test many inbreds. And our first stage of that for most of our programs now is with genomic prediction. And then we're running two more stages, 2 more years of field trials where we're reducing down those number of inbreds until we have a core set of inbreds that we think will work well for commercialization. And then from there in a hybrid program, you're testing what are the ideal hybrids that that you're going to test. So at each year, we're in that first funnel for a hybrid program, we're picking which inbreds advance. So that's like a general combining ability. And in that last stage, we're looking for a hybrid program at what is the specific combining ability. So what's the specific hybrid? For an inbred breeding program, it's actually a similar process, but you don't have a kind of a second funnel, but we're really looking at more like very careful fit for where to place that inbred. So in the beginning part of the program, there's still a like kind of overall performance. And then as we get closer and closer to commercialization, it's what is that final fit for that variety. And then the second kind of level of decision making is when do we pick parents that are going to get recycled. So there's 2 arrows in this diagram. The first is picking the parents that are going to become the parents, picking the inbreds that we're going to use as parents to generate the next beginning funnel. And then which parents we're going to enter into trait integration. Most of our breeding programs in North and South America are selling traded material. So that has biotech traits. And we actually are doing almost entirely our breeding with conventional non-traded germplasm. And so we go through rounds of backcrossing, and there's a decision point on like which entries we're going to put into that trait integration backcross program. And for regions where we may not be selling biotech traits, we still use this trait integration backcross program to integrate native traits, disease resistance, other traits that are like QTLs that we may not have in that germplasm that we want to help commercialize, that's going to help it to have a better fit. So even in Europe, say in France, where we may not be selling a biotech trait, we may be introgressing like northern resistance through TI. And so you have decision making to make at each year for what advances to like through this funnel. And then you have decisions on what things you're going to recycle back as parents and what things you're going to recycle to commercialization as a traded or a native trait improved variety. The other thing in thinking about timing is you have the timing that we are selecting for like testing. So you have a testing cycle for a given region that mostly is one testing cycle per year. There are parts of the world where we can get 2 testing cycles in per year. For example, in Brazil, there's a safrinha market, and then there's a like summer market. So in Brazil, you can have 2 testing seasons of corn per year, but it's really different germplasm in those 2 different markets. So it's effectively like still about a 1 year. You have a decision point you're making each year on what's getting reduced in this funnel. But your nursery cycles for us are not running on that same time scale. So most of our programs are on 2 cycles per year, or 3 cycles per year, or even 4 cycles per year. I think the fastest we can go is in canola, where we can get up to 6 cycles per year in growth chambers and in greenhouse. But so you have decisions on making populations that are turning at a faster time scale than the decision on what's getting advanced for testing. And the way that those interact is we're mostly developing the selfing or DH first, then we're running selection with genomic prediction or field testing. And then from there, you already have fixed inbreds. And so you're using multi-cycle to make hybrids for the next stage, or to increase seed so that we have enough seed to commercialize. So if you think of like a soybean variety, we don't have DH, so we're doing like inbreeding up to like F4, and then F3 or F4, then testing F3 or F4 derived bulks. But we have to be able to increase enough seed to commercialize at the very end still. And so we're still using multi-season nurseries to be able to increase seed quickly to get enough seed that we're going to commercialize for those varietal crops. And for hybrid crops, having multi-season nurseries allows you to have, fresh seed available always for that next year's hybrid testing. And so you always can test new varieties, new hybrids on time. I think what else here that I wanted to point out in that cycle. So some of these decisions are dependent on performance in that year's yield trials, and some of those decisions are independent of that. And so if we can make a decision that's independent of the timing of when we have like an advancement, so we can spread that out through the year and spread the scope of the workout. So like genomic prediction is something that, we update the model once a year, but you can make selections throughout the year, which helps us for the lab because we can spread out when samples are going to the lab. So we have starts for new populations that get made throughout the year so that we can smooth out that nursery work, and we can also smooth out what samples are going to the lab. And so you can manage the kind of ebb and flow of the program that way. For other things that are dependent on this decision on how performance from field trials, most of the time between when the field trial is harvested and we need to make an advancement decision is very short. And that's going to come into play later when I talk about analytics, because say for corn trials that I'm most familiar with from all those years working on corn breeding, we would be harvesting, if the advancement meeting, say, is on a Wednesday, we would be harvesting up till Sunday. We would get the data on Monday, look at the data on Monday and Tuesday, and make advancements on Wednesday. And then decisions for what goes to the nursery would get made that same Wednesday, and they would be packaged and shipped by Friday. So the amount of time between getting data, QCing that data, analyzing the data, and making the decision is really tight. And that changes kind of how we think about managing that data. And it changes how you think about decision making. And is there ways that you can kind of prepare for that decision to make that decision easier and faster, so that when you do get that data, that it can quickly be translated into action. The other thing I will say about this is, if you look at this program and you think of, okay, you're getting data every year and you're making decisions every year for nursery and for hybrid or varietal advancement. The other thing to remember is this data is going to get used as training to predict future years performance of a variety. So you're accumulating data as you're getting further and further in the program. And inbred, as it gets multiple years of testing, you get higher confidence in its performance. But you're also using that data from multiple years of testing to predict new populations that may be related or descendants from those inbreds. And so it's important to think about that data life cycle as like, it's not single use. Like, you use it once to make a decision for advancements that may be like really short and quick, but then you have kind of decisions that get made with that data that may really be coming even years later. And so how that data is stored and how you think about QC is really important, because if you're going to be reusing that data point to make decisions over and over again over the next 10 years, then it's really important to be able to connect the performance data to the genetics data and to the context where you're going to make that decision again in the future. So, what do we measure? And, what are we using to make that decision? And I have two figures here. One is from the University of Illinois. That's like a kind of a description of the different growth stages for corn. And the other is a screenshot I took from our website where you can order seeds. So if you go to Dekalb and search for hybrid corn and you want to buy seed, you can search for what seed is for sale in your maturity, and you can find performance from the late stage trials that are run on strip trials, like on maybe other customer fields or on our fields. But you'll also get this like descriptor of what things that commercial and sales thinks are going to convince you to buy this particular hybrid. And this is a really early hybrid that's also dual use for silage and for grain. And you can see the things that like sales thinks that a customer really wants to know about this hybrid that's going to make them want to buy it. And really what we're trying to measure in the breeding program is the traits that are going to help us essentially to fill out this performance list. It's going to be measuring the traits that are going to help us differentiate our performance against our competitors. And it's also the traits that are going to help us to understand how well this particular hybrid or variety is going to work for production, and it's going to work in customer fields. And so as we start from the beginning, you measure traits that may impact like germination, because that matters to when we sell. We may be measuring traits as we get later and later in the corn life cycle, they get closer and closer to grain harvest. And what you're trying to do is match what is economically the most important to your customer and to our production, but also the things that we know as scientists that are important physiologically that may help improve our prediction for those other traits. So even though you may measure something that is a correlated trait, or we may measure it in a different way than our customer may see it, but that measuring those like correlated and related traits helps us to get closer and closer to what we know that we need to measure for being able to sell the variety. And when we're thinking about like what is the decision point on what things get advanced to in the next stage, it's really around how does that particular hybrid or variety compare to the internal checks. So we'll use the current commercial varieties as our checks, or we'll use inbreds or hybrids that are like key to beat or that have key traits for breeding. But we'll also look at, say, our competitors and say, okay, if we need to sell a corn hybrid against a Corteva or a Syngenta variety, what's the advantage that we need to be able to have to be able to differentiate ourselves in the market? And so and you can look at that across all the different crops, and that even if it's a veg crop, it's like, what are the things that maybe that help drive adoption of this particular tomato variety, and maybe its flavor profile, maybe its shelf life for a processor. But those are the things that you have to take into account as you're making decisions in the breeding program. We also do use, in a pretty integrated way, genomic information throughout the advancement process. So at the very beginning, we'll fingerprint pretty much every parent that we use for crossing. And then every F1 that we're going to advance gets checked to be able to make sure that the parents we think it has are the parents it really was used. We'll check for heterozygosity. Essentially, you're looking for mistakes. And we'll also check the inbreds that we're going to put into the yield trials to make sure that the inbred that we're going to put into trials and test is really the inbred that we think it is. And those are usually pretty inexpensive markers that it's not that many markers that we're running to do that, but the payoff is really huge. Like, we find even with the best controls and the best nursery management, it's pretty common to find 5 to 10% of the F1s or the inbreds are either had an outcross or a seed mixture, or something's happened where the seed is no longer pure. And that can really mess up your data, especially if you think about using that data repeatedly in building a training set, for example, for genomic prediction. And so it's really important that for us that we use fingerprint and these quality control markers. Then we're going to run marker assisted selection. There's probably 2 different levels that we'll do that. Sometimes it's for specific haplotypes or QTL that's with very few markers that just track that specific piece. And sometimes that's using genomic markers that are covering the whole genome. And we aren't genotyping that for genomic selection at the same density that we're fingerprinting. We're using imputation to be able to then impute up to the level of fingerprint, even for the entries that we're selecting at lower density. And the reason for that is really economic. So if someone was willing to sell us like fingerprints, millions of fingerprint chips for the same price as we can genotype for genomic selection, then heck, we'd buy those. But right now we have in-house genotyping that's at a lower density that we can do really cheap and really fast. And then we impute up to fingerprint level, both for training and for selection. And then we have, for discovery and validation, we use a lot of custom markers and a custom genotyping to be able to try to discover new trait associations. And for validation, that can be anything from few markers that are flexible to like genomic sequencing. It really depends on what the project is and what it needs. So for genomic selection, the main way that we're using it is to eliminate a year or 2 years of our testing program. So if we can get the same prediction accuracy as the first or the second year of testing, then we're going to jump the material that we can select with genomic selection to the next stage. That next stage of testing, if once you've implemented genomic selections, not only is important for predicting new things that are going to advance in your testing program, but become really essential as the way that you keep your genomic prediction model working so that your prediction accuracy stays good. So it's again this way that you're reusing data over time. Is you have to think about that first year of testing not only as your first chance to see performance in the field, but as a way that you're going to keep your genomic selection predictions for the next 2 or 3 years as accurate as possible. And what that has allowed us to do is, because field testing is more expensive than genotyping, we can reduce that amount of field testing, then increase the size of the program. So effectively, you have a much bigger program, even though your field footprint is actually smaller. The next stage of this, like, what are we deciding, is you're really deciding what is the future plan. So when something's advanced, say you take something from genomic prediction and you advance it to the next stage of testing, you're really advancing that with a data plan, like a data package that says where it's going to be tested, what's the experimental design, what are the comparisons that you're going to make, whether that be checks, whether that be commercial hybrids, so that the next year you can make decisions on where it goes in nursery and then what the experimental design is for the next level of testing. And then the other thing you're deciding is what are the analytics that you're going to apply to that, and what kind of pool of data you're going to use it for in the future. So who's making these decisions? So our breeding program, this is from, this slide may be a little bit old actually, but you know, we're spread out around the globe. The row crops, we have 6 main crops that we work with. We also have a sorghum program that's, but it's a joint venture. So I'm going to leave that out with it and of this discussion. But for these 6 crops, we have sites all around the globe. Breeders are spread out around the globe, but we also have kind of clusters where we have more scientists. So like St. Louis is probably is a big hub. Germany now is a big hub. And then in Africa and Asia, South Africa is probably our biggest hub. And then in Asia, we have a lot of people in Singapore, but also in Bangalore, in India, in Pakistan, and then we have a joint venture in China. And then South America, our biggest hubs are really in Brazil and in Argentina. And I put this one, I added this one in. This is not a corporate slide, but it looks like a corporate slide because it just has like kind of team structure. And I wanted to put this in for 2 reasons. One, it shows what this product design team, product development team, it kind of explains what my role is and our team's role. But I think it also shows something I think really important about like how we think of making decisions in this breeding program and how we've divided up the groups that are in and how like centralization has affected this. So like the product design organization is that beginning part of the breeding program. So making decisions about what crosses we're going to make, that inbred development, the selection with genomic markers, and then that first year of testing. Product development is going to be, once it's advanced out of that first year of testing, all the way to commercialization. And their focus, so if you look at how those are divided up, the product design teams are divided by crop, and they have they don't have a regional assignation. So cotton, we're mostly in the United States, but for example, my team also thinks about commercialization, what populations we're going to make for Brazil, for Europe, for Asia, and for Africa. The corn team is spread out across the globe for product design, but in development, they have a very regional focus. And the teams in North America really don't care about performance in South America. They care about interactions with the customers and the commercial teams and the local needs. So I think the reasoning behind this is to like have one team that's thinking global and another team that's thinking local, and that there's this like tension between them where the local teams is always like keeping that focus on the like, what do we need specifically for a county in Iowa in North America, or for a departamento in Argentina. And they may be thinking across different crops. So in that county in Iowa, they may be planting corn and soy. And the team the people that care most about that county are going to care about both corn and soy. Whereas the geneticists and the plant breeders that are on my team, for example, are just working on cotton. And so that, but we may be thinking about cotton globally versus locally. So that's how that division gets made. This is a recent org structure for us. I have a slide on this later, but the one sure thing in Bayer and Monsanto is that like org structures that we have will not be the org structures we will have. So if 3 years from now I present again, I can almost guarantee you that this structure won't be what it is. But for now, this is how we think about organizing this like global versus local. And then the other thing is we have a number of enabling teams that are centralized that work across crops and across regions. So planning, that's helping to plan not only resources and like budgets, but also where we're going to have nurseries and where we're going to have yield trials. The testing team is executing yield trials and measuring the traits on all of our testing through the from the beginning stage to the end. Analytics is doing analysis across crops and across regions. Multi-season nurseries are running, you know, in multiple locations around the globe. And then we have a group that's strategy and operations, and you know, you can think of that as like HR, finance, all those other support things that, you know, really make everything go. And then I have a dotted line, and then these other green boxes is really the other parts of the company that have input or are involved in the decisions that we're making in breeding. And that is a broad range from regulatory, chemistry, biotech, production, commercial, which includes our sales organization. So all of them will have different input at different stages of the breeding program. One of the like books that was like all the buzz here at Bayer and Monsanto, it was one called Team of Teams by General Petraeus. He was a general in the US Army, and he wrote this book about different kinds of team structures. One that's this traditional top-down structure, one that's like kind of distributed decision making, and then this other one that's like team of teams where you have like independent teams that can make decisions independently. And he argues that this team of teams is the like ideal organizational structure. Sometimes people at Monsanto or Bayer will tell you that's what we have. It's ambitious. I don't think that's really our team structure, but the idea of it is the essence of it, I think, is still a good one, in that what we're trying to do is making sure to enable teams to make decisions independently, but also make sure those teams are not just from one organization. So, for example, in product design, we're in the end responsible for making all of the new populations for the cotton program. But if we make populations for the cotton breeding program that aren't fitting what the development team is seeing and what the customer wants, then we fail. And so the decision, like within our team, we're actually kind of intermixed on who's making decisions on what populations we're making. It's a shared responsibility between people that are on my team in product design and that are in product development and that are in the enabling teams. And we really do rely on each other to make those kind of decisions. I alluded to this earlier, and I think actually I could have probably given this whole talk with just this one slide, because this is the one thing that's probably the biggest factor in what makes Bayer different, for example, from the company I worked for before, which was Ag Alumni Seed, that was a much smaller company. We changed at a much slower rate. Change is pretty inevitable here. Like I broke this up into kind of 4 different time periods. The Dekalb Holden days is probably like late 90s to the like mid 2000s, so like 2005, 2006. Then I would think of this like molecular Monsanto, really like Monsanto combining the different companies and the cultures of those companies that they had acquired in the late 90s into a cohesive breeding program. I called that the molecular breeding program. That was probably, say, 2006 to about 2013. And then in 2013, we rebranded it internally as like breeding 3.0. Maybe those other two stages are breeding 1.0 and breeding 2.0. We never called it that internally, but we just skipped to 3.0 because it sounds cooler. And then this next phase, because we didn't want to go to breeding 4.0, apparently, the brand of that is precision breeding. And what's the same and what's different with those? I tried to outline like both some of the like really practical, like small changes. Like, I mean, they're relatively small. So like think about breeding software in the Dekalb Holdens period, like this breeding 1.0, we had local servers at each station with local software. The breeding software we used then was either Virgo or Maris. We had two different softwares depending on which kind of breeding program you had. Every breeding program kind of did it their own way. It was local databases. It wasn't stored centrally. And that's when they just first started using like BLUP, for example. And it was every breeder used that differently, analyzed their data differently, and then advance things quite independently. So the strategy then was really to have kind of competing programs that were like the Dekalb type of program and the Holdens type of program. Then in this molecular breeding phase, we kind of we merged the germplasm between those two different, like the different companies. So now you had like a common germplasm source. We have a common database that's centrally like managed, and we have breeding software that's also like centrally managed that was called Midas. And you know, Monsanto invested in this large computing cluster that was in St. Louis. And if you look at like our promotional things, they would even show pictures of this, like that it was in this building that was like you could hit with a tornado and that the computers would be safe. And then if you think of the innovations that come from, you know, the kind of innovations that made that different from before is we did have centralized analysis that came, automated analysis. We were using molecular markers really consistently in those time periods, but it wasn't genomic prediction. It was individual QTL that we would map within a population, and then we would chip seed and so increase the frequencies of those. And then the other thing that allowed teams to do was to increase the size of the breeding programs a lot. So that was the big strategy shift with that. And then in breeding 3.0, it wasn't really a size increase, which was the strategy. It was using genomic prediction. It was maximizing our use of DH and chipping for corn, inbred, like single seed descent for our other crops. It was using OR and predictive analytics as well to help make decisions. There was also a shift in our software. So we moved off of Midas to Apollo and Velocity. Velocity is the newer one that's now replaced Apollo. Both of those are web-based tools that are run out of AWS. Our data also moved from this central cluster to either AWS or Google Cloud, depending on the data type. And then in this next phase that we're really just entering is we're fully transitioned now to Velocity. So it's a web-based breeding software. The databases are Product 360, and those are entirely housed on AWS and Google Cloud. And then the breeding methods, if you think of what does that mean different from before, it's moving things faster. It's using like more complex models. It's phenotyping with advanced phenotyping. It's moving from an OR model to something that gets closer to AI. And this is really what we're building now with the new teams. And so hopefully that works. What I can guarantee you is this will not be the end of Bayer reinventing how we run our breeding programs. Like that will continue. We have teams that are devoted to like thinking past precision breeding to like what comes next. And that was my old team, and like we will change. Like whatever the breeding software is today, I can guarantee you there'll be a new one. We may move off of AWS and Google Cloud to something else. Like, and I don't know what that is, but change is built into the plan. Make sure I'm on time. I'm a little over so far, so I'll speed up. This was kind of a description of what was BrIN 3.0. If you think about it, was a focus on genetic diversity that was centralized. So using global diversity for and moving germplasm around the globe, making sure that we understood native traits that could be used across regions. We did a lot of seed movement. So if I had a good inbred in the United States, it was also used as a parent in Brazil. It may have been used in Thailand. It may have been used in China, in Africa. And that's part of why we have all of our breeding programs running on native traits, I mean, without biotech traits, because having biotech traits in our forward breeding stuff would have prevented this seed movement. Uh-oh, my battery is running low. A sec. Hopefully I can have enough to make it to the end here. So then breeding methods, analytics. So what is different with precision breeding? It's making our TI, trait introgression, which I'll try not to use acronyms, but it's hard because I use them all day. To run faster, it's making decisions faster. It's shortening the time of our breeding program. If you think of that funnel, it's ways that we can shorten that funnel, that we can think of running population development faster and making decisions using prediction that may be something that we can make like 3 times a year versus once every 5 years. So can we cycle faster than we were in the past? It's improving how we're using data science and artificial intelligence. You can even see that in hires that we've been making. We've invested a lot in people that have data science skills, and they're getting more and more integrated into the running of our breeding programs. And it's in how we run our operations. It's how we think of validation and using global data and connecting that across stages and across regions. So, and it changes how we think about operations. So this picture here is our seed processing facility. This is counting seed and randomizing seed and then putting it into trays for it to get planted. And this facility here will count essentially all of the seed for North and South America and put them all in trays and it will all get planted, where that used to be all distributed work that was happening at each station. And if we think of how that's changing our testing footprint, we're thinking about how do we test if we are going to try to place our product with more precision with the customer. How do we use our testing footprint to do that? So testing in more diverse conditions, thinking about how we use that data to predict customer performance as well as to predict earlier to speed up our breeding program. And then like if you look at all the data points that go into like how we're making decisions, for example, about which crosses to make, there's a lot of potential crosses you could make, millions of those. And what you're doing is predicting which ones have the highest chance of success. And then we're going to cut down even further by looking at native traits. So maybe it's disease resistance, QTL. We're going to look at genetic diversity. So we're going to use that fingerprint data to compare like whole genome diversity to try to maximize the performance and diversity of the entries. And then we're going to be looking at performance. I think there's another one in there that I can't read, but essentially you're using all of your historical data to reduce that down to the populations that we think are going to have the highest chance of success. And the type of data that you're combining together, and I think this is an important thing if we think about databases and IT, is you want to be able to connect genotype data, phenotype data, and environmental data together to be able to run models that includes all three and can be run across historical data sets so that you can go back as far as possible and kind of maximize the utility of that data. And because what you're really using is you're like reusing that data multiple times to be able to predict what's the best populations, what are the best entries to put into testing, and what's the right way to test them. And all of these models kind of work in a similar way. BLUPS is essentially this, right? We have a testing set that you're going to so that you measure in the field. You're going to predict tested and untested entries. The learning algorithm for BLUPS is essentially this mixed model. Some of those models can be linear models. Some of them could be machine learning models. If you think of BLUP this way, you have the mixed model learning from the field data that you're testing, and then you're predicting genotypes that you may not have tested or relatives that you may not have tested, or you're predicting entries that you have tested in environments that they may not have been. And so that's where you use that to generate decisions. Sometimes we can even do those in terms of a probability. So if you do that as like a random forest model, you can get a probability of getting success, but some of those are like a fit. It's like BLUP, you get an accuracy and but you don't necessarily get a success probability. But all of these we're thinking of the same kind of way where you're taking what is the historical data, running picking some subset of that historical data to be a training data set and a test data set to be able to understand the accuracy.
Thank you very much, Brian. I guess very likely.
I think actually we must have lost Brian because he just mentioned that he's running out of battery. Okay, okay, so I'm gonna save. There were a bunch of questions in the chat that I am going to save. Some of them are proprietary information. Some of the questions touch proprietary information, so I think Brian can't answer all of them, but I will pass the questions on to him, and whatever he can answer, I'll ask him to like write up a short blurb and send it out to the same mailing list. The seminar is recorded, so and we'll be sending that out as well. So yeah, that is that is unfortunate. We had 5 more minutes to have maybe some Q&A, but I guess yeah, technology. So sorry everyone for this abrupt end to the proceedings.
Yes. So Gary, I saw you have some comments. Would you like to make a comment about the 2-stage final breeding cycle?
I could make a couple comments. I thought one of the things that was very interesting about the talk was what he didn't talk about. He didn't talk very much about breeding cycle time, but it's pretty clear that Bayer is implementing a rapid cycle 2-stage genomic selection process.
Yep.
That is very similar to what was outlined in Chris Gaynor et al. In the Crop Science 2017 paper. And one of the best lines of evidence for that is they hired Chris Gaynor and John Hickey and the authors of that paper who have been doing the simulation for them. So I imagine that touches pretty closely on proprietary information. But really, you know, and we're getting very similar information out of the excellent simulation work that the EIB is doing, using the same tools and the same approaches, showing that accelerating the cycle and really separating out our selection for breeding value, the way we think about making the decision about parents for advancing populations, from selection for commercialization. There are 2 steps in the process. So that, you know, it would be nice to hear his comment on that, but he wasn't talking about it. It's interesting.
Yeah, I think so, so that sort of stuff can get a little touchy in a present. Oh, Brian, you're here.
I'm back. What am I not talking about, Gary?
Oh, Gary wanted you to talk
Rapid
more about
cycle,
cycle time,
two-stage breeding. Yeah,
Yeah,
cycle time and simulation. Yeah.
so that's essentially like this like division between product design and product development is to like
Mhm.
make that a more prominent part of our breeding programs. So like that's one of the big parts of my new team is doing rapid cycle essentially for cotton.
Mhm.
And John Hickey's team is doing rapid cycle for corn. And it's a little bit making, yeah, like kind of your this two-part program is we have the design team doing product kind of population development and the development team doing product development and selection of hybrids and varieties.
Can you talk more generally about how simulation influenced the design of Bayer's program?
So, and Young Wah could talk about this because when she worked here, she helped develop a lot of that capability. So we have probably two different ways that we think of using simulation. One is like simulating changes that we would make and understanding kind of impact on genetic gain or on logistics. So we may like simulate like new, like kind of increasing or decreasing something and then see what the change is there. Then we would we have also very specific simulations where we would simulate like genotypes that we currently have, say in population in product development, and we'll simulate kind of the way that we would do TI. So with the specific inbred and what are the inbreds that we could pick as donors and see which donors have the highest chance of success. So that simulation is using like very specific, like known genotypes, whereas the other ones you can simulate like 10 years of a breeding program and say, okay, is this breeding program going to give me the same amount of success for the same dollar amount? Or we simulate it and say, oh my goodness, this is going to mean we need to have 100,000 nursery plots and we only have capability to do 90,000. So, what do we change then in the simulation to fit the logistical like constraints that we have?
Mhm. All right, we're actually at time. Thank you, Brian, for a fantastic talk.